Next, we'll take a look at how to perform a rigid body refinement using PrimeX. To perform a rigid body refinement, we go to the Selected Task button in the PrimeX toolbar and choose Reciprocal Space Refinement. The Reciprocal Space Refinement interface appears, and from the Refinement Method menu, we'll select Rigid Bodies. When performing a rigid body refinement, we typically recommend choosing the Truncated Newton Minimizer under the Minimizer drop-down menu. Next, we'll select all atoms to define the rigid body group by clicking on the All button, which causes the phrase All to fill the selection field. Once we've selected all atoms, we can click on the Add button to create the rigid body group. We're now ready to run the rigid body refinement, which we'll do by simply clicking on the green Start button here and submitting the job. At the conclusion of the calculation, the output structure is incorporated into the project table and appears in the workspace, where we can see that the R and R free values have both decreased slightly. In this example, we can perform the rigid body refinement again, but with different groupings to get a better result. First, let's hide the protein side chains using the Undisplay button in the Maestro toolbar. Next, we'll rotate the structure in the workspace, and it's easy to see that there are two domains that we can use as two separate groupings. Note that in many multi-domain proteins, such as kinases, each domain is a contiguous group of sequential amino acids, and we could define the groupings for rigid body refinement simply by supplying two ranges of residue numbers. In this case, though, if we color according to residue position using the color palette item in the Maestro toolbar, it's immediately apparent that this isn't possible here. Fortunately, it's still a simple matter to define the two groupings. Let's go back to the Reciprocal Space Refinement panel. First, we'll click Delete All to remove our previous rigid body group from the table. Then we'll make sure that the Pick toggle is activated and tell Maestro to select entire residues when we pick atoms in the workspace. Next, we'll click and drag to lasso all of the atoms in one lobe. Clicking on the Add button will confirm our selection and define the first lobe as a rigid body group. Finally, we can invert the selection for the first lobe in order to define the second lobe. To do so, we'll click on Select to bring up the Atom Selection dialog, and then click on Invert to invert the selection. The second lobe is then highlighted in the workspace, so we'll accept the selection, and then click on Add to create the second new rigid body group. To run the calculation, we can click on the Start button and submit the job with a new name. At the conclusion of the job, the results are once again automatically incorporated into the project table and displayed in the workspace, and we see yet another decrease in the R and R free values.